Hello everyone, welcome back to my channel. In previous section we had studied about principles of genetic engineering. Now in this section we will be knowing each principle in detail. The first principle was creating rDNA or recombinant DNA. Let us now learn what is recombinant DNA. Recombinant DNA is nothing but when foreign DNA or gene of interest is combined with vector, we call it as recombinant DNA. Foreign DNA or gene of interest is which character we want from a particular organism, that particular DNA we are going to extract. After extracting it, we need something to get into the host. For that, a vector is required. Vector is also a particular DNA sequences which will be serving as molecular vehicles. So when you take this particular DNA sequence of a vector and then foreign DNA or gene of interest, they will combine together giving us the recombinant DNA. Here there is no involvement of host yet. We are just seeing the foreign DNA combining with the vector, that is recombinant DNA. So what is a vector? Vector is used to transfer genetic material into target cells. So target cell is nothing but the host. Helps to replicate and express the inserted DNA sequence into the host cell. So whatever character we want, that can be expressed into the host cell only with the help of a vector. This is gene of interest plus vector DNA which will combine to form recombinant DNA. So we know that to insert gene of interest into the host, we need vector which is compulsory. So let us know different types of vectors that are existing. First one is a plasmid. So what is a plasmid? A plasmid vector is a small circular extrachromosomal DNA molecule. So it is a DNA molecule but extra chromosomal. So apart from chromosome, there is some other molecules which are present which we call it as plasmid, and it is small and circular and which replicate autonomously. It can self-replicate. Auto means self, it can self-replicate. So it is a small self-replicating circular and extra chromosomal DNA molecule. So if this is the bacterial cell, we will be having chromosomal part here. So apart from this, there are some small circular DNA molecules which we call as plasmids. They have region where gene of interest can be inserted and introduced into the host. Example, antibiotic resistance gene, okay, which is our gene of interest, plus plasmid DNA which we obtain. Together they will be forming a recombinant DNA. Later this recombinant DNA can be introduced into the host, that is for example E. coli. Apart from that, we have cosmids which are used in molecular cloning again. Then we have artificial chromosomes which are BAC and YAC. The abbreviation is bacterial artificial chromosome and yeast artificial chromosome. So these both are used when large number of sequences are used. For example, bacterial artificial chromosome has nearly 3 lakh base pairs and yeast artificial chromosome has nearly 2 million base pairs. So these are used where we are going to obtain the large number of sequences. Then also we have bacteriophage lambda and M13 as vectors. Here comes the very important part. Stanley Cohen and Herbert Boyer are the two scientists. They constructed first recombinant DNA in the year 1972. So how did they do this? They cut a piece of DNA from a plasmid carrying antibiotic resistance gene in the bacterium Salmonella typhimurium. So Salmonella typhimurium is a bacteria. It has a plasmid. In that plasmid there is a particular region where it is resistance for antibiotic. So that region is extracted and linked to the plasmid of Escherichia coli. So Escherichia coli is also a bacteria which has a plasmid. To this plasmid, this gene of interest is inserted. So now the Escherichia coli, which wasn't so much resistance to antibiotics, can now show much resistance for antibiotics like chloramphenicol, tetracycline, streptomycin and sulfonamides. So in order to achieve all this, there are three basic steps which is involved in genetically modifying an organism. The first step is identification of DNA with desirable genes. Previously we saw an example, right, that we needed an antibiotic resistance gene in E. coli. So if we need that, we have to take an organism Salmonella typhimurium and we have to extract the desirable genes which will code for antibiotic resistance. The next step is introduction of the identified DNA into the host. Now we know what to do. We have to take a plasmid and with help of plasmid we are going to insert the gene of interest and then introduce them into the host. So what happens after we introduce them into the host? We should maintain the introduced DNA into the host and transfer of the DNA to its progeny. Maintain in the sense we have to give the proper conditions so that this cell will not get destroyed or disrupted. Then it will start to multiply and transfer of DNA to its progeny. For its further generations, that character should pass on. So all these three processes will be achieved with the help of enzymes. Now we will see what are the different types of enzymes that are involved in such process.